Hello, my name's Tim Sheehan, and I would like to talk about the kinetochore structure and its function. Now, to start with, I would like to talk about the structure. The kinetochore, which is all of this, is located on the sister chromatids, the chromosome and its copy. The kinetochore itself is a relatively large complex of proteins. It's about 38 different types, all in all, and it's about 100 nanometers across. The only thing that even comes close to this complex is the ribosome. Now, onto the structure itself. The inner plate, which is right here, houses a very interesting histone protein called the CENP, PA. And the CENPA replaces the H3 histone protein, which handles genetic regulation in the chromatin. Now the next structure, which is the outer plate right there, this actually handles microtubule attachment. It's got 20 anchoring sites in an animal cell. And a prokaryote, it generally houses just one. Anyway, this is the fibrous corona. This little thing catches and holds the microtubules and is responsible for adding and holding the tubulin on the microtubule itself, microtubule being here. And that basically wraps up the structure bit, so in my next part I would like to talk about mitosis and how this all comes together. Alright, now that I've talked about the primary structures, I would like to take a moment and talk about the microtubule's control and how it weaves the chromosomes into position. This is so mitosis can occur within the cell. The first step is interphase. This is where the cell is simply living. It's not doing much in the way of growth, in the form of reproduction. In this section, the chromosomes cannot be identified individually, but are a huge clumpy mass of DNA. You can see this with the nucleus right here, called chromatin. Interphase is also where the cell starts to duplicate its internal structures. And it is also where the chromosome starts to pinch itself and the centromere can be seen. I can't show that here, but more importantly, the structure that assembles the kinetochores, the MCAK, the mitotic centromeres, associated kinesins, construct the kinetochores here in this interphase. The next phase is prophase. This section is where the cell condenses and chromatin can be seen individually. This is also where the centrioles, right here, they are reproduced. This was done during interphase, and they make up the base for the dividing of the cell. The nucleus also falls apart during this phase. Okay. Now that we've reached prometaphase, some things kind of start to happen. The centrosomes start to migrate to both opposite sides of the cell. Now, the kinetochore fibers, right here, attach to the kinetochore. Thankfully these are magnetic, so this shouldn't... There we go. Like that. And the kinetochore starts to remove tubulin. And so it would move toward here. Now, things are being... The other centrosome will then hit here as well. and attach to the kinetochore. Stubborn little thing. Anyway, once this is done, they will add and hold tubulin until the kinetochores have reached the middle part of the cell. This ends prometaphase. Alright, now we have met metaphase. Now, metaphase is basically where the sister chromatids are both lined up in the cell, right in the middle. And this can go on for a while. It depends on the cell, varies. But once this is done, we eventually reach anaphase. Now, anaphase is where the kinetochore tubules pull the sister chromatids apart. Now, there is something in here called the stem bodies. Now, the stem bodies signal the cell that it's time to divide. Now, we finally reached the last phase, which is telophase. Telophase is mostly where the small little parts of the chromosome become small little nucleuses. These fuse into larger nucleuses, hence here, and it signals the cell to divide. Now the dividing factor right here would be like this. Right here would be the cleavage furrow. Now, the contractile ring that is in here is made of myosin and actin. And these are like muscle fibers, and they pull on each other until the cell contracts, making, as I said, contractile a furrow. Now, this goes for a little bit, and the plasma membrane adds on until we have two separate cells, two separate and new daughter cells, which ends mitosis. Alright, to make a quick, to make this presentation end, I am going to basically do cell death in my own way. So, here you go, last treat. Magnificent. Anyway, that's all I've got for you guys, so, signing off.